Yes, hello. My name is Russell Whitaker. I'm a software engineer at Google in Mountain View, California. I had the distinct pleasure to welcome back Dr. Aubrey de Grey, a research gerontologist at Cambridge University, from whom we had the pleasure of receiving a talk last month hosted by Jonas Carlson. Without further ado, Dr. de Grey.
There's so little ado that I haven't quite got my microphone on. There we go. Marvelous. Thank you very much, Russell, and for that introduction and for having me. Yes, it's a great pleasure to be back. A month ago or so, I gave a rather general overview of the work that I do and that I coordinate with regard to seriously combating aging. And there was quite a lot of interest, but of course there wasn't nearly enough time for me to tell you about anywhere near everything that we do. So I thought that it would be good to be able to go into some more depth in respect of one of the most interesting and most complicated components of this work, which is the approach that I think we need to take to combat cancer. Cancer is, of course, something that kills people, and it kills people with a higher prevalence the older they get, more or less. So it's something without which it would be of limited value to fix everything else in aging, and that's why I regard it as part of my job, so to speak. This is going to be a good summary of the philosophy of what I have to say. It's a good summary of the philosophy of how I think about combating aging in general, in fact. I think that a lot of the more conventional approaches that have historically been followed in this area have tended to oversimplify the problem of combating aging, or in some cases actually to undersimplify it, to conclude wrongly that nothing is possible simply because it's unimaginably difficult. And yes, well, Einstein was usually right, and I think this was a good example. So the talk will be structured like this. I'm going to talk, first of all, a little bit to amplify the remark I made a moment ago about cancer being an important part of aging and how it fits into the whole phenomenon of aging and the effort to combat it. Then I'm going to talk about the general structure of how I think we need to combat it and why the approach makes sense. You will see during part two that it's quite ambitious and that there are an awful lot of things that might make it rather difficult. So I'm going to go through some of the more obvious versions of that in the third part of the talk. And I may have run out of time by then, but if not, then I will go on and talk about some perhaps slightly less intuitive pros and cons of this approach. So starting here, well, those of you who were here last time will probably recognize that I put up a table something like this, which I spoke about a little bit during my talk a month ago. And essentially this is the summary of how I think we can break down the problem of combating aging into sub-problems that can be tackled not exactly independently, but at least individually. The problems are listed on the left-hand side and the solutions on the right-hand side. And the solutions, as you can see here, are very much ideas to repair and reverse the accumulating damage of aging that is listed on the left, as opposed to merely slowing down the continued accumulation of those problems. Oh, I'm being given another laser pointer just as my own one has run out. Thank you very much for that. Let me just find the right button. It's probably this one. It is. Yeah. OK. Actually, I think I'll point at this one. So in my last talk, I gave a relatively brief overview of the sixth of the seven things down here, junk, indigestible molecules that accumulate inside cells and which lead eventually in old age to phenomena such as atherosclerosis and neurodegeneration and one or two other things. Today what I'm going to focus on is the bottom one, mutations in our nuclear DNA, our chromosomes, and the ways that we might cope with that. Now, I'm going to start by amplifying the very short description of the problem here. I'm talking about nuclear mutations as opposed to mutations in the mitochondrial DNA, which I consider useful to regard separately. But I'm not just talking about mutations. I'm also going to be talking about epimutations, and I'm going to be explaining what an epimutation is in a moment. And then I'm going to be justifying this rather outrageous claim at the bottom, which is that cancer is really the only consequence of nuclear mutations, or epimutations for that matter, that we actually need to worry about. So if you remember from last time, I spoke about the possibility of actually giving people who are already in middle age before we start the prospect of an unlimited extra healthy lifespan and essentially indefinite youth, which is something that I've been calling engineered negligible senescence. And I spent some time last month on the concept that we may be able to achieve this very far in advance of actually achieving the whole of the technology that delivers it. The reason this is true is because of a concept called longevity escape velocity, which I described in some detail last time, so I won't talk about that now. But the upshot is that if we can do these various things well enough to give middle-aged people as little as 30 years of extra healthy life by this panel of bona fide rejuvenation therapies that actually repair and reverse the accumulation of various molecular and cellular changes, then that will be equivalent, for functional purposes, to a completely comprehensive, perfect rejuvenation therapy. So the one I'm going to concentrate on, as I said, is this bottom one today. First of all, what's an epimutation? Well, first of all, I'm going to say why we need to worry about epimutations. It's a simple thing. Different cell types, of course, express different proteins and different RNAs in order to have a different phenotype, a different behavior. Different cell types need to do different things just in order to be useful components of different organs. But of course, all of our cells, more or less, have the same DNA sequence. So this is not done by changing the sequence. This is done by changing something else. And what it changes are the epigenetic characteristics of our various genes. So an epimutation is like a mutation in the sense that it is an accidental, adventitious alteration, but it's not an alteration to the sequence. It's an alteration to these decorations that determine which genes are expressed and at what level. Methylation of the DNA, especially of cytosine bases, is the best known aspect of epigenetic regulation. And other things that happen are modifications to histones, the proteins that the DNA is wrapped around. Various different types of modification happen to histones, including methylation and phosphorylation. And then at if you like, a higher level of abstraction, a higher level of, a higher order of organization, there are changes to the density, the tightness with which our DNA is packed into chromosomes. So all of these things can suffer these accidental, adventitious changes, and all of those changes collectively can be referred to as epimutations. That was a term coined perhaps 15 years ago by the famous gerontologist Robin Holliday. Now, the idea that mutations, and indeed epimutations, could only matter in respect of cancer is a pretty ambitious one. And one thing I probably ought to elaborate on is that I'm not actually really saying they're the only thing that matters. I'm saying they're the only thing that matters that is not already covered by other aspects of the strategies for engineered negligible senescence, the SENS plan. Certainly, nuclear mutations and epimutations cause damage that induces cells to undergo cell suicide, apoptosis. So you get cell death, and in some cases, that cell death is not compensated by the natural cell division of other cells to replace the cells. So you need to do something about it, and that's one of the major categories within the SENS plan. That's what stem cell therapy is for, basically. There's another phenomenon that happens, which is cells get into a state where they're supposed to die, but they don't any longer listen appropriately to the signals, either from inside themselves or from neighboring cells that are supposed to be telling them to die. And that, again, is a separate category within the SENS plan. There are a variety of techniques now being developed to essentially encourage or remind cells that they ought to be listening to signals to commit suicide, and that's going rather well. So these two things are definitely going to happen, and I'm not saying they don't matter. On the contrary, I'm saying they do matter, but they are covered. The question is whether anything else matters, apart from cancer, in a currently normal lifetime, in terms of the accumulation of nuclear mutations. And I'm using the phrase here, general sort of non-specific cellular malaise. The thing is, of course, that if there were a very generalized decline of cell function as a result of spontaneous accidental mutations and epimutations, we could be in real trouble because different cells would have different problems and would therefore require different solutions, and we have a rather large number of cells. So we could have a rather large number of problems and rather a large number of required solutions, and that would be very difficult. Whereas here we have more or less two rather well-defined families of problem that are altogether more straightforward, albeit not very straightforward, to address. So I want to explain to you why I think we don't need to worry about general cellular malaise. And it's always good when putting forward a radical interpretation of existing data to be able to point to two things. First of all, to the actual data that seem to support one's contention. And secondly, to be able to explain from a theoretical basis why the contention actually does make sense, even though one might not have realized it beforehand. So here's the data. And this comes from the laboratory of Jan Weich in Novato in Marin. And in fact, this is the group probably that's done the most work on the potential for non-specific general cellular malaise to result in a normal lifetime from nuclear mutations and epimutations, and to lead to, therefore, contributions to aging. Now, some tissues accumulate mutations throughout the lifespan. This is in mice, of course, which is why the x-axis goes up to something in the region of 3 years. The small intestine is an example of a tissue in which mutations do genuinely accumulate, albeit it seems at a decelerating rate. There are good reasons why we don't need to worry about the small intestine, and that's something that I'm going to come back to later in the talk. So just hold that thought for the moment and concentrate instead on the lower part of the graph, especially on the very lowest. The green line here is the trajectory, the age-related trajectory of mutations in the brain. And as you can see, mutations rise in frequency during the very early part of life when the mouse is growing. And this is easy to understand because cell division is a very mutagenic process. It's error-prone. Sometimes replication of the DNA simply throws in incorrect bases. So that's no surprise. But throughout adulthood, the abundance of mutations in the brain goes up by an undetectable amount. That's pretty damn good news, because something that contributes to aging, by definition, it has to change with age. And if it's not changing, then, therefore, it probably doesn't contribute to aging, which is pretty good news. But why does it make sense? Why would we have evolution? Why would evolution have given us such good DNA repair and maintenance machinery that there was no increase, to speak of, in the abundance of mutations in the most important tissues, such as the brain? Well, this is a very interesting problem, because we generally believe in gerontology that our anti-aging intrinsic systems, in other words, our repair and maintenance systems, tend to be only as good as necessary. But this seems to be an exception. And so what one needs to do is explain why it should be an exception. And this is my explanation. And incidentally, the paper in which I describe all this just came out literally last week, so you can go and get it in Mechanisms of Aging and Development at the moment. There is a very well-established phenomenon that was, in fact, first proposed about 50 years ago by a famous evolutionary geneticist in America called George Williams, called antagonistic pleiotropy. The idea he had was that genes, or indeed genetic pathways, that are beneficial to the organism early in life will be selected for and retained during evolution, even if they are actively bad for us late in life. And this is simply because there are not so many organisms around in the wild of an older age than there are of a younger age, because, of course, shit happens. Organisms get eaten, they starve to death, they freeze to death. So the selective pressure to respond to the good or bad consequences of the possession of a particular genetic pathway is lower if that pathway is expressing this good or bad consequence late in life than if it is expressing it early in life. And if it's doing both, as I'm describing here, then the young phenotype will outweigh the old phenotype. This is not controversial at all. This is completely accepted by gerontologists, and possibly the most extreme example is something that I'll actually come back to later on, the interdependence of cell proliferation potential for the maintenance of tissues like the blood or the skin that need to be continually replenished by stem cell division, as against the potential for uncontrolled cell division and the need for pathways that control cell division, which is, of course, something that we need in order not to die of cancer. Now, what I realized is that a very similar concept can be applied to the comparison between mutations that cause cancer and mutations that cause general cellular malaise. And I'm calling this particular concept protagonistic pleiotropy because what we're talking about here is genes or pathways which are good in two ways. And let's suppose they're good for one reason, I'm calling it reason X, early in life, and they're good for another reason late in life. Now, for exactly the same reason that antagonistic pleiotropy works, it's also the case that genes of this sort will only be retained because of the reason early in life. The fact that they have this additional beneficial effect late in life will not be the reason they are selected for. And that means that the defense against this late life problem, in other words, the ability to avoid the late life problem, will indeed appear to be what you might call unnecessarily good, which of course is exactly what I was showing you here. This lack of accumulation of mutations in the brain is the demonstration that our ability to avoid accumulating mutations in the brain is unnecessarily good. All right, so this is pretty good news. We have a theoretical understanding and an experimental understanding of why general cellular malaise will not be likely to accumulate as a problem that contributes to aging in a currently normal lifetime. I've gone on and talked a little bit more about what the mechanism for this is. Very straightforward. The reason why cancer-causing mutations are special is because it only takes one cell to kill you if it gets the wrong sort of mutations that cause it to divide uncontrollably and eventually become a life-threatening cancer. Whereas for anything to matter at a biological functional level to the organism that does not involve loss of control of the cell cycle, it has to affect a lot of cells. It has to affect a large proportion of the cells in whatever tissue is supposed to have that function. So basically, anti-cancer defense is a requirement that evolution has experienced, and it has to drive the quality of our nuclear DNA repair and maintenance machinery up to a point that is unnecessarily good with regard to general cellular malaise. It's had to do this purely in order to avoid us dying of cancer before we have had any offspring. That is the whole the protagonistic pleiotropy concept applied to cancer. Now, those of you who know a bit of biology may have appreciated already that this is a little bit oversimplistic. Here is something that we need to bear in mind in this. There is a lot of redundancy that is built into cancer-specific defenses. We have lots of different ways to make cells effectively detect that they have undergone mutation or DNA damage and simply to commit suicide or else to go into a state called senescence where they won't divide as a defense against becoming cancerous. And there is also the fact that these defenses only apply to cancer, as well as the fact that they are numerous and mutually redundant. All of these things essentially offset the argument I just gave about the cell number, the fact that it's only one cell versus a large number of cells. But this is not a complete argument unless we can actually suggest that they totally offset the numerical disparity argument that I gave a moment ago. And we can see quite easily that they would not be so good as totally to offset it by simply asking ourselves, well, let's see what would happen over evolutionary time to an organism in which this whole concept, this protagonistic pleiotropy concept, did not apply to distinguish cancer from other mutations. If we have an organism in which all mutations matter equally, whether or not they cause cancer, then there are going to be a variety of anti-mutation mechanisms, whether they be stopping mutations from happening or repairing them or whatever, and they will fall into these two categories. There will be defenses that are specific to cancer, and there will be defenses that are against other sorts of DNA damage that could cause general cellular malaise. Now, repair and maintenance machinery that applies to the whole genome, by definition, combats both of these things. It combats mutations in all genes. That means that it will be more strongly selected for, both for its evolution and for its retention, than the pathways that only defend against cancer. Therefore, what's going to happen is that the selective pressure to maintain these pan-genome repair and maintenance pathways will be higher, and mutations will accumulate less easily over evolutionary time. Therefore, you will get a progressive, essentially a progressive victory of the pangenome repair maintenance mechanisms over the cancer-specific ones. The cancer-specific ones will certainly be retained because they have a good deal of utility, but they will not be the only ones that are retained. So the defenses against general cellular malaise will end up being unnecessarily strong. In other words, protagonistic pleiotropy will emerge. That's the theory. Now, I emphasize, of course, at the bottom that it is only a theory, and I would be the first to concede that we need a lot more data on all of this. The experimental work that I showed in a few slides ago is one of the few studies that have been done that really thoroughly address this thing. And it's only been done in one species. It's only been done in one lab. You know, other experiments have been done that are related, but not nearly enough of them. We need more work on this. But that's the general principle. So coming back to the whole theme of this part of the talk, and to basically conclude this part of the talk, I just want to explain how cancer fits into the aging phenotype, the aging problem. And in particular, I want to explain how it will fit in as and when we develop the therapies that will be necessary to address other aspects of aging. Of course, many of you will be, have only limited, if any, knowledge of the various approaches that I propose to attack other aspects of aging. But most of you will know about some of them. I mentioned stem cell therapy briefly earlier on as something that is required to replace cells in areas of the body that do not naturally replace their cells. And I'm sure you all know that in some areas, stem cell therapy is moving rather well, even to the clinical trial level. Similarly, other areas of biology are bearing down very effectively on a number of the other types of molecular and cellular damage that are side effects of being alive in the first place and that eventually cause us to die. But the point is they're going well. And so if we were to neglect cancer, we really could, with quite high probability, end up in, let's say, 20 or 30 years from now, in the rather invidious situation whereby we can fix everything else, and we have increased our lifespan by a little bit, our life expectancy, but only a little bit, and then we'll all die horribly, because we'll all be dying of cancer, because we dragged our feet and didn't get on and invent cancer therapies that were as effective in terms of postponing death from cancer as other therapies were for postponing death from other causes. So I don't regard that prospect as particularly attractive, and I think that gerontologists in general are beholden to consider cancer as part of their problem, which a lot of them don't. So what is my solution? Well, as I said, first of all, I'm just going to really give an outline, and then I'm going to drill down a little bit into some of the more obvious problems. Here's the outline. So cancer is the hardest part of aging to combat. I've told you one reason why that is true, namely that just one cell going AWOL can kill us. But the big reason why cancer is the hardest part of aging to combat is because it has natural selection at its disposal. It is a furnace of genetic inventiveness. In fact, every cell in a cancer is a furnace of genetic inventiveness, and there are, after all, 1 trillion cells in a cancer that's big enough to be clinically relevant. So we have the big problem that if you give some sort of therapy against a cancer that broadly works, then there will be, with high probability, some cells in the cancer that have mutated, or indeed do mutate in response to the therapy, to resist this therapy and to survive and withstand it. And unfortunately, of course, having withstood it, they will then proliferate, just as the original cancer did, and you'll be back where you started, except that the cancer will be even harder to get rid of than it was before. This is a big problem, and it's a particularly big problem simply because we've got an awful lot of DNA to play with. One approach to addressing this rather daunting prospect is that we could just apply lots of cancer therapies all at the same time, so that the cancer just had an awful lot of problems to solve, so to speak, and maybe it just wouldn't succeed. And that might work some of the time. But I think it's useful to try to think way out of the box here and to look at the prospect that there might be a cleaner, more if you like, monolithic solution, but nevertheless one that really, really works well, and furthermore we can see why it would work, and we can work towards, if you like, the rational design of the implementation of that therapy. And a particular thing that I want to point out is I really meant out of the box. I mean that it's okay for a solution of this sort to be so ambitious that in today's world we wouldn't want to consider it. In a world, in other words, where other things kill us in an age-related way already, where other things become more problematic with age. What we're looking at here is something that we must look for therapies that might not make sense until and unless we fix everything else in aging. So here is the essence of my proposal. It seems to me that even though evolution is very clever, and evolution within a tumor is very clever, there is only so much it can do in the time that it has before the cells just wither away and die. There are only 1 trillion cells. You may think that's a little bit of a strange use of the word only, but it's still a finite number of cells. And when cancers mutate to resist therapies, and indeed to resist the anti-cancer mechanisms that the body has intrinsically, what they tend to do is they turn particular genes up or down, or on or off. Genes that, for example, will break down some particular chemotherapeutic compound, or will chuck it out of the cell before it gets to a sufficient abundance. Things like that. What they don't do, what cancers do not do, is invent whole new genes out of thin air. They sometimes, of course, mutate genes so that they just don't work at all, which is a sort of special case of changing gene expression level. But creating a new gene out of nowhere is something that cancers just don't do, because it takes too long. It's too unlikely in the context of a cancer. Evolution does it all the time, of course, but evolution even doesn't do it very often. Evolution mainly works at the level of changing gene expression. And the point, evolution has a lot longer. Evolution of the organism has a lot longer and a lot more cells to work with than the evolution of a cancer. So this is the proposal that I have been putting forward over the past few years, whole body interdiction of lengthening of telomeres, another of these rather mouthful names that I seem to be rather prone to give to things. And here's the general concept. Well, actually, no. Before I give you the general concept, this is, for the non-biologist here, a little bit of a piece of background on the role of telomeres in aging, because this is probably the aspect of the biology of aging that is most profoundly misunderstood by the general public. And indeed, worse than that, it's an area that the general public think they do understand. So telomeres are the ends of chromosomes. They are sequences that play a role, first of all, in stopping chromosomes from being joined together end to end by the machinery that mends broken chromosomes. But secondly, they're there to solve something called the end replication problem. Our DNA, our chromosomes, are, of course, molecules that have ends. They are not circular, unlike bacterial DNA, which is circular. And when the DNA replication machinery works on a linear DNA molecule, it doesn't complete the job. This is a very profound and intrinsic property of all DNA replication enzymes, is they are unable to complete the replication of the very end of the chromosome. And this was realized back in the early 1970s, actually, independently by a Russian professor and by Jim Watson, who is famous for one or two other things. So there's this problem. The chromosome gets shorter, a little bit shorter, when it's replicated. So, of course, when cells divide, they have to replicate their DNA. And over the lifetime of the organism, whether it be a mouse or a human or whatever, there will be a certain number of cell divisions in any given cell type, and the chromosome will thereby get shorter. A lot of people, a lot of non-biologists, think, well, this might actually have something to do with aging, because as cells continue to divide, they become progressively unable to divide any further because of this problem, because their telomeres go away and they start doing things like, indeed, joining chromosomes end to end. And they end up, therefore, with a problem. Now, it turns out that that's not true, or at least it's very nearly not true. Most cells simply don't divide that often in the lifetime of the organism. They have plenty of telomere, enough telomere to get them through. So they just don't have this replication limit on them. Furthermore, there is an enzyme called telomerase, which compensates for the end replication problem and re-lengthens the telomere. And telomerase is, of course, very important in the germline, in the cells in our ovaries and testes that create sperm and eggs that lead to the next generation, because, of course, over one generation to the next, there is an indefinite amount of cell division that needs to be maintained. But telomerase is also expressed at trace levels, but at adequate levels, in all cell types that we have that have to divide more often than they could with the amount of telomere that we're born with. That basically means stem cells of tissues like the blood, or the gut, or the skin, or the lung, which divide reasonably regularly because the cells that they give rise to are constantly lost. In large mammals, only the cells that need to divide a lot of times in a lifetime express telomerase. In mice, which for this purpose counts as small mammals, there is an awful lot of telomerase in most of the cell types. But in humans, we only express the absolute bare minimum of telomerase in the cells that need it. And that seems to be true for most large mammals. And this is well understood. This is well understood to be a really great defense against cancer. Because if you're a small mammal, then you've got to have a bit of telomere in your cells just to keep the cells happy and not joining their chromosomes end to end. But even a little bit of telomere is enough for a cancer to grow, even without telomerase expression, without this compensatory mechanism, to grow enough to kill the organism. But if you're a human or a cow or a gorilla, then you can withstand a tumor that's, let's say, an inch across. It's not going to kill you. And if that's as far as it can get without doing something about its telomeres, then you've got much less of a problem. If a cancer has to turn on telomerase in order to carry on dividing enough to kill you, then you better use that. It's something that you can use to your advantage to avoid getting cancer. So that's why large mammals, which would not be killed by a cancer that was small, only by a cancer that was so big that it couldn't get that way without expressing telomerase, they make sure that the cancers do actually have to mutate to express the telomerase. In other words, they turn telomerase off in all the cells for which they can get away with that. So this means that the general concept that is present in the popular understanding of the biology of aging, namely the concept that cells get into a state where they can't divide anymore because their telomeres are too short, is only a very rare phenomenon in the human body. It happens in a few cells here and there in a lot of tissues, and it happens in one particular tissue quite a lot, the immune system, which I don't have time to talk about today. But it's a relatively rare phenomenon compared to what most people tend to think. All right, why have I told you all about that? Because I want to tell you what the therapy is. This is WILT in a nutshell. The idea is to take cells that from the human body and manipulate them in just the same sort of way that is very much in the news at the moment, to de-differentiate them into a more primitive state, like an embryonic stem cell, and then to re-differentiate them into the state that we'd like them to be in. In particular, in this case, we would like to get them into a state where they were faithfully behaving as stem cells of each of our various rapidly renewing tissues, like the blood and the gut and the skin. Of course, this is something we can't quite do yet. We're getting there, though. There is an awful lot of work going on to make this a reality. What I propose is that we actually eliminate the genes for the compensatory mechanisms that I mentioned earlier on. I've mentioned something called ALT here. I'll come back to that later, so don't worry about that for the moment. But in particular, to get rid of the genes, actually not simply to suppress their expression, but to eliminate the genes themselves using ex vivo genetic manipulation. Of course, we need to make sure that these stem cells have respectably long telomeres initially, so that there can be some use. I'll probably be able to get to this other last thing about genetic resistance to chemotherapies. That's a bit of a detail, really. But the idea here is that if someone's got cancer, or even if, once we get this working safely, they are old enough that they might get cancer soon, then we can introduce cells like this into the body, and they can act as our stem cells. Now, of course, stem cells, by definition, they divide and differentiate to make tissues that matter. And as I mentioned, stem cells of these sorts of tissues have to express telomerase in order to last all our lives. So these cells will not work forever. However, they probably will work for about a decade. So a periodic replenishment of our various stem cell pools with cells that do not have any telomerase genes and therefore could not mutate to compensate for their end replication problem, this would be a way of maintaining our stem cells, our stem cell pools rather, without the individual cells, but not giving us a risk of cancer. Quiescent cells, cells in tissues that are not maintained by stem cells, are amenable to a similar approach, which in this case would be using in situ, in vivo gene therapy rather than genetic manipulation in the laboratory. But the same concept applies. OK, so that's all a bit of a mouthful. So let me show you what I mean graphically. The situation we have at the moment in someone that doesn't get treated for cancer is they have various continually renewing tissues, like the blood and the skin, which continue and renew indefinitely throughout the lifespan of the organism. But if a mutation or a sequence of mutations happens that makes something a cancer, or nearly a cancer, starts it to divide uncontrollably, then eventually, when it gets into a particularly mutagenic state called crisis, it will turn on telomerase and thereby be able to divide indefinitely. And that will cause the cancer to grow a lot and eventually to become malignant and metastasize, and that's the end of you. Now, therapies that we have at the moment for cancer are all characterized by this lower half of this slide. Namely, the same sort of thing happens, and when we notice the cancer, we do something about it. We try to knock the cancer back and kill the cancer without doing ourselves too much damage elsewhere. But of course, because cancer cells are just cells and they are rather similar to other cells, there's only so much we can do, and so we may knock the cancer back a little bit, but by and large, we won't extend lifespan all that much. Supposing we just didn't have telomerase, supposing I was able to click my fingers and just eliminate the telomerase genes from all your cells, well, you wouldn't die of cancer, because a cancer would get to this highly mutagenic state, and that wouldn't be enough. It wouldn't have a telomerase gene to turn on, so the cancer would eventually regress before it kills us. And any new cancers that came along later would do the same thing. But it would all be completely irrelevant, because you'd be dead by then, because you wouldn't have any blood, because your stem cells would have given up. So WILT proposes that we do exactly this. We eliminate the telomerase genes, but we also periodically replenish the stem cell pools in this way, so that when our stem cells are giving up because of the end replication problem, we rescue the tissue by putting in new stem cells that still don't have telomerase genes, but do have the sufficiently long telomeres to keep the cells going for about a decade before they get to have the same problem and we have to do it again. Well, this is a rather ambitious idea, and the more you know about biology, the more aghast you will be at what I've said so far. I'm quite sure of that. But I feel that, as I said earlier on, we do have to take cancer seriously in order to have a prayer of coming up with therapies that will combat cancer enough, as much as we will be combating other aspects of aging within the foreseeable future. And so I'm rather reminded of another physicist's remark, Niels Bohr, who said this. I think it was to Wolfgang Pauli at some point. Well, what I'm going to do for the rest of the talk is to try to give you a feel for why it may be crazy enough, and also it may not be too crazy. I ran a workshop back in 2004 to, well, actually, no, the workshop was in 2002, to evaluate this. This was a proposal that I first came up with only in March of 2002. And the very first thing I did was to convene a nice little invitation-only workshop at which the world's leading specialists in all of the various areas that may bear on the plausibility of this approach came and considered the whole thing in great detail for a whole day. And they were sufficiently convinced that it wasn't obviously not going to work, that they were willing to all sign up to a paper that I wrote, that I drafted anyway, that was published about 18 months later. So that's a good start. This is definitely not just me coming along and telling you that something might work. This has authoritative support and endorsement from other people. So what are the more obvious problems, and what are our chances of overcoming them? Well, as I say, I'm going to start with whether this is actually good enough, whether this is enough to really outwit the ingenuity of cancer. The first question you may have is, well, hang on. Cancers, as I just said, are composed of roughly 1 trillion cells by the time they kill us. Now, 1 trillion is only 2 to the power 40, okay? And that means that a cell can go from being one cell to being a trillion cells in only 40 cell divisions. Now, in fact, telomeres, even in humans and other species that have relatively short telomeres and no telomerase in most cells, telomeres are long enough to allow considerably more than 40 divisions, probably more like 60 or 70. So it looks like we might be in a bit of a problem. But it turns out not to be true, for two reasons. The first reason is that there's not just cell division in a cancer, there's also a humongous amount of cell death. Cells are mutating all the time, as I mentioned, and most of those mutations are not good for the cancer cells at all. Most of them are absolutely fatal for the cancer cells. So there's a fantastic amount of cell death going on, which of course increases the number of cell divisions that the cells that don't die need to do in order to get up to this level of a trillion cells. Second thing that means that what's called the Hayflick limit, the limit on cell divisions, is indeed our friend when it comes to cancer, is that the mutations that cause cancer don't all happen at the same time. What will happen is one mutation will happen that will knock out one particular pathway and allow the cell to start dividing. But after it's divided a little bit, it'll find that it's being constrained by the extracellular matrix. So it has to do some more mutation that causes genes to be expressed that break down the extracellular matrix. And those second mutations don't happen in all of the, let's say, 1,000 or million cells in this aspiring cancer. It'll only happen in one of those cells. So let's say it's got to 1 million cells after 20 cell divisions. After 40 cell divisions, it won't be at 1 trillion. It'll only be at 2 million, or in probably 1 million, in fact, because the early cells will have died. And of course, it's not just 2 mutations that you need to get a cancer, it's quite a lot. So anyway, the upshot of all this, to cut a long story short, is that most people reckon that a cancer needs to get up to at least 300 or 400 cell divisions before it becomes fatal. So that's pretty good news. Fallacy number 2. Telomere shortening is dangerous. Well, this is true. So I mentioned earlier on that when telomeres get too short, the consequence is that chromosomes start to be joined together end to end. And that's very, very bad for the cell. But initially, the cell might just survive because the cell has ways to break chromosomes apart again. They don't necessarily break in the same place, mind you. And lots of things can happen as a result of that. In particular, lots of genes can become either present in only 1 copy rather than 2, or only 3 copies rather than 2, things like that. So you have, of course, thereby a lot of changes of gene expression, which is exactly what cancers need to do to mutate to do well. So indeed, it turns out that having really short telomeres is bad. It's mutagenic. But that's only bad for the initiation of cancer, for the progression of cancer to a point where it might need telomerase. In order to get beyond that, you've got to have telomere maintenance, because the overall effect of this mutation process, even if it involves joining chromosomes together and breaking them again, will kill the cell after not many cell divisions at all, after only a few at most, unless it can find some way to stabilize the system by expressing telomerase and repairing the ends of the chromosomes again. So I said I was going to mention ALT. I haven't mentioned ALT yet. Telomerase, it turns out, is indeed activated to high levels for this purpose in perhaps 90 or more % of cancers, in virtually all cancers in the blood or the skin or other tissues that are maintained by stem cells. But in some tissues, especially in skeletal muscle, for example, which leads to sarcomas, or in the brain where you have glioblastomas, or actually liposarcomas in the fat tissue, in there, up to half of cancers do not actually express telomerase. They've invented a new system called ALT, alternative lengthening of telomeres is what that stands for. And we have a problem. We don't know which genes are expressed in ALT that cause it to be able to do this. We know that the dynamics of telomere lengthening is very different in ALT than it is in telomerase. It's much more chaotic, but it works. And it's rather important that we figure out what genes are running this system. We haven't got it yet. But the good news is there must be some genes that are activated in ALT and not present most of the time, as opposed to the opposite, where it's just sort of a loss of the functioning of genes that are always activated. We know this because inactivating genes is much, much, much easier than activating genes that have been deliberately turned off by methylation of DNA and so on. And we know that because telomerase activation in cancers that do have telomerase is a very late event during cancer. So There are going to be some genes. We just have to find them. And lots of work is going on this now. It's fashionable. So for the purposes of today, you can regard the problem of WILT as being equivalent to the problem of telomerase. OK, so that's my reasons for thinking that this will work if we can do it. And I want to spend the last, I suppose, 10 minutes on the question of whether we can actually do it. Whether we can, first of all, implement this, remove the telomerase genes from enough of our cells to make a difference. And secondly, whether we can implement the fixes to the various side effects on stem cell dynamics that I mentioned earlier. Let's do that second one first. Well, the thing is, some of this is actually already rather easy. Let's look at the blood. There are lots of diseases of blood stem cell maintenance, aplastic anemia and dyskeratosis congenita and things like that. And the treatments for them are not exactly working perfectly yet, but they're getting there. They're not bad. And those therapies simply involve a bone marrow transplant. It involves replenishing stem cells with new stem cells that are working better. And bone marrow transplants, you know, we'd like to be able to do them better than we can, but we can now do them pretty well. So this is technology that, by the time we've fixed all the other really rather difficult aspects of what I've been telling you, we really ought not to have a problem with. Now, the blood is not the only tissue that we need to worry about here. I mentioned earlier that we need to worry about the gut, the lung, and the skin as well. That's probably all, incidentally, but nevertheless, we do have to worry about all of them. It's been done in mice and dogs already in the gut, essentially either using straightforward surgical techniques or using chemicals that essentially strip away the gut lining so that you can replace it with other cells. It's not been tried in humans. It's a little bit not ready for prime time, but progress is being made. Again, in the lung, the same sort of problem exists. Cystic fibrosis is a disease of the lung which could, in principle, be treated by stem cell replenishment, and so that's being looked at. And the skin, interestingly, is going very well this way, partly because of the medical problem of treating burns, treating serious burns, and secondly because of straightforward cosmetic surgery, people having chemical peels to rejuvenate their skin. This shows the sort of thing that actually happens and why it's not too hard to replenish the stem cell pools in the skin. The skin, to a first approximation, has two layers, the dermis and the epidermis, and it's only the epidermis we need to worry about here because the dermis has cells in it that do not divide very often, cells called fibroblasts. It's the epidermis that matters, and luckily it seems that the dermis orchestrates the behavior of the epidermis. So this is a figure from a paper in which a colleague of mine removed the epidermis from some normal hair-forming skin in a mouse and replaced it with some epidermis from the cornea of the eye, which is obviously a very different sort of epidermis. And within no time at all, like a matter of days, or a week or two anyway, that epidermal tissue had been reorganized and reprogrammed by the underlying dermis in such a way that it behaved exactly like normal endogenous hair-forming epidermis, just like this, and hairs were indeed formed and so on. So this is rather good news. It basically means that the stem cell maintained component of the skin is so plastic that it can be essentially directed to do the right thing by cells that we do not have to replace. The gut is an interesting tissue for one other reason, namely that according to what you might read in the academic literature, the stem cells in the gut divide quite often, a lot more often than the stem cells in the skin or the blood. And that would be a problem here. It might mean that rather than replacing the stem cell pools every 10 years, we might have to replenish them every, let's say, month. And that would be altogether more impractical. However, there are some good reasons to believe that this can't possibly be true. Probably the best reasons come from studies of mice whose telomerase genes have indeed been removed, not by gene therapy, but by germline manipulations in their parents. These mice do not get problems in their gut any sooner than they get problems in blood and the skin and so on. So this means that by one mechanism or other, their stem cells are maintaining telomere length for the same amount of time. They have that same rate of telomere loss as other tissues. One interesting possibility is that they do this not by having a slow rate of cell division, but by segregating the strands of DNA appropriately so as to keep the long strands in the stem cell. This is a model called the immortal strand hypothesis that's had some very interesting experimental support recently. We don't know, but the thing is we don't need to know. We just know that the gut does not have a problem that is more serious than the blood or the skin. I mentioned earlier that some tissues that get cancers are not maintained by continuous division of stem cells, muscle, brain, fat. And here we have this problem of WILT, but we also have the problem that we can't simply maintain these tissues by changing the stem cells outside the body. We are going to have to do somatic gene therapy. And furthermore, it's harder than normal somatic gene therapy, where you just put a new gene into the chromosome. This is a case of something where you want to change a gene, in fact, in this case, delete a gene or two that are already present in the chromosome. That's a technology that goes by the name of gene targeting. And there are two major ways to go about it. One is to exploit our own repair machinery, to basically trick our repair machinery into incorporating changes into the DNA when, in fact, it's trying to incorporate restoration of what it perceives as damage. And the second approach is to use a very clever system that bacterial viruses, bacteriophages, have evolved. And I'll come to that in a moment. First of all, homologous gene therapy. Basically what happens is you put in some DNA that looks like a bit of damaged chromosome, that the DNA repair machinery recognizes, or rather misrecognizes, as a problem. And what happens is that by essentially a process that's similar to what happens during meiosis, during cell division making sperm and eggs, you get the new DNA, the engineered DNA, incorporated into the chromosome in place of DNA to which it is very similar in sequence, in other words, homologous. And this is a very cool way of achieving these sorts of approaches. Unfortunately, precisely because it's a damaged repair process, it's not very reliable. This DNA will preferentially go into the place it was designed to go in, because these green bits on the engineered DNA are similar to the green bits in the chromosome. But with an unacceptably high probability, it will also go in elsewhere, or in multiple copies, or anything like that. So I have high hopes for an alternative approach, which is this bacterial virus-mediated approach. Bacteriophages have special sequences called attractor sites that allow them to integrate into the genome of the bacteria that they have infected. And they do it not by hijacking the machinery of the bacterium itself, but rather by carrying their own genes for this, genes called integrases. Integrases are very sequence-specific. They allow a recombination event, a crossing over event, that incorporates these two circles into one circle. And because of the way they work, combining two sequences that are not identical to each other, they create a situation here in which the same enzyme does not do the reverse reaction. You need a different enzyme called an exogenous to do that. So this is a lovely way of regulating the system. But also, because the phage is a piece of circular DNA, rather than a linear piece of DNA like on the previous slide, the whole business of adventitious, unscheduled insertion of this DNA into a random, wrong place in the genome is much, much rarer. It hardly ever happens. So this is a much more reliable and controllable way of going about this sort of thing. And I have high hopes that we may be able not only to insert, but to disrupt genes using this technology. I suppose I have a couple of minutes left, but I wanted to leave time for questions. So perhaps I'll just stop here and let people pipe up. Thank you.
So if anyone has any questions, here's a microphone. Do you have a question? Yeah. Okay, sure.
Aubrey, as you know, I agree with a lot of what you have to say, but you didn't talk much about the fact that stem cells rather than somatic cells are, I think, emerging as the responsible for cancers, and also the role of telomerase is much more multivariate than just the lengthening of telomeres. So obviously, if you get rid of it, you're going to, in effect, affect an awful lot of other things.
Dear me, I seem to have a slide on this. So certainly it seems clear that stem cells are the origin of a lot of cancers. And this is really no surprise, especially for cancers that are maintained by the expression of telomerase. because if a cell has its telomerase genes really, really heavily, completely turned off, then it's going to take a lot of mutation and epimutation to turn that gene on again. Whereas if it's sort of slightly on already, because the stem cell needs it to divide enough, then it's going to be easier. It's going to take less mutation to turn it on to a sufficient extent to maintain the cancer. So this is the main reason why we think that stem cells are largely the main problem that cause cancer. Cancers have, well, there's a concept that's very popular these days called the cancer stem cell, which is a slightly different thing. This is, within a mature cancer, the subset of those cells that seem to have a lot of division potential. But that's a separate question that we really don't know what that has to do with the cancer. We do know, however, that whether stem cells are responsible for cancer or not, and whether stem cells exist already in a mature cancer, they still have to divide, and therefore they still have the end replication problem. So it doesn't really affect what I've been telling you. Now, what might very much affect what I've been telling you is that, as Kevin just mentioned, the roles of telomerase may not be restricted to telomere lengthening. Now, I do want to say may, because actually it's very hard to determine for sure whether a particular phenotype is caused by telomere shortening or by some other deficiency of telomerase if you do an experiment in a telomerase deficient context. However, a couple of questions that we really need to ask in determining what impact that would have on WILT are these ones at the bottom. First of all, telomerase is actually not just one protein. It's one protein plus one RNA molecule, which is encoded by a different gene. And the evidence that we have at the moment that there may be roles for telomerase outside the lengthening of telomeres, that evidence predominantly, in fact, overwhelmingly relates to the overexpression of the protein component. So we have the option, in other words, to knock off the RNA component and achieve the WILT goals and leave the protein component alone to do whatever it might do. The second point that I think really has to be drawn attention to is that the protein component, which is the one that seems to have some interesting roles in mice, that have been found in mice, the protein component is the one that is suppressed in human somatic cells as much as possible. The RNA component is actually expressed quite prolifically. So it looks very much as though whatever problems mice may have that they solve by expressing a lot of telomerase, we have already found, our evolution has already found alternative solutions such that not allowing cells to express any telomerase at all would not have an effect. Now, of course, this is a relatively weak argument. We certainly need more experimental data on this. It could be, for example, that stem cells do actually need their trace amounts of telomerase, even though they're real traces. But at this point, I would say things are looking good.
So I've just been told by our AV tech that he is graciously allowing us to run over a bit. So we're not as compressed as we would have been otherwise. So feel free to use this time, and we'll field more questions.
Sure, yeah. Any other questions?
On to Chris Phoenix here.
So what percentage of cancers depend on being death resistant? And so if you cure death resistance, how much does that solve the cancer problem?
Unfortunately, not very much at all. Cancers essentially aren't death resistant at all. Cancers have a vast amount of cell death all the time, simply because death resistance requires particular pathways to be working, as well as particular pathways not to be working that would allow them to be death sensitive. And so mutations are happening all the time that just make a cell not work at all. And it may not die cleanly by apoptosis, the nice, neat cell suicide mechanism. It may die just by running out of steam and keeling over in a rather dirty way, but it'll still die. And the only reason cancers are a problem is because cell division is so much more potent than the cell death. So no, it's really a very separate problem.
So you mentioned replacing stem cells, which is possible in some systems, but more difficult in others. How about some near-term applications like inhibit siRNA or even telomerase vaccines, which are closer to term clinically to approach these sorts of issues in the shorter term?
Yeah, I'm all for the extensive pursuit of therapies that are easier to do than WILT. And no one would be happier than me if it turned out that we could come up with a therapy that was as effective as WILT could be, but was much easier to implement and much less scary. But at the moment, I don't think it's going to happen. If we look specifically at ways of combating telomerase expression, then ultimately they have the same problems as ways of combating any other aspect of cell growth. Number one is that some cells need their telomerase, so you've got a therapeutic index problem. If you express too much inhibition, then it affects your stem cells too. Number two is that any small molecule that you throw in, any chemotherapeutic agent that happens to specifically target telomerase, can be combated in the same way that other chemotherapeutic agents are combated, whether it be by breaking it down or by excluding it or by upregulating telomerase expression in the first place to compensate, all the usual things that happen. So I'm really pleased that work is going on in all of these avenues, and I very much support it, but I don't think we should bet our lives on it.
Anyone else? No questions?
Chris has got one.
So it sounds like cancer cells kind of universally need telomerase.
Telomerase or ALT, yes. They need telomere maintenance mechanisms.
So I've been hearing about anti-tumor therapies that, for example, look for, is it niacin receptors on the cells? And I'm wondering about the possibility of detecting cancer cells and piling into them with chemotherapy drugs based on simply the fact that they produce telomerase.
Yep. Chris asks an extremely useful question, which is, can we not simply identify cells that are expressing really high levels of telomerase and use that as a way of getting rid of them, rather than by reducing their ability to express telomerase, simply by killing them as a result of the fact that they express them? And that's certainly already being tried. Indeed, it looks as though the body probably tries itself to do this, because the immune system is an extremely important component of our inbuilt anti-cancer therapies. I think that would be possible in principle, and if we could do it really well, it might even work, because, of course, the reason why telomerase vaccines, for example, or telomerase inhibitors might work is that cancer cells are dividing so much faster than stem cells that you could get away with a longer treatment, so to speak. However, the problem is that if something is controlled by, if it's expressing the right protein, subject to the presence of such and such a drug or whatever, the problem is that it could mutate so that it no longer is dependent on the drug to express the protein. And that's exactly what you would expect to happen, unfortunately, rather quickly. Because the drug the drug dependence sequence would be a sequence that could simply just be deleted or could be eliminated by a translocation that put the telomerase coding region under a different promoter. And these are the sorts of things that happen all the time in cancers.
Actually, the question was more along the lines of, suppose your alternate telomerase used a different component to extend the DNA itself. So it's not a matter of regulating the expression of telomerase. It's that you have a telomerase that's using something other than conventional bases to extend the...
I think the same applies, except that you have the additional problem that the whole telomere maintenance system driven by telomerase is actually a much more complicated system that doesn't just involve these two genes. It also involves a whole bunch of other telomere-associated proteins. So, for example, if you if you had this alternative version of telomerase that put on a different sequence, then that would cause a load of other problems for the chromosomes and for cell division and so on, let alone if you tried to put something other than DNA on. So, yeah, but over and above those two things, still the same thing applies, that the drug dependency of the expression of the telomere maintenance mechanism would still be something that could mutate into no longer being dependent, and therefore you wouldn't have your solution anymore.
You've discussed various biological approaches to addressing the telomerase issue and cancer in general. But I'm curious, a lot of these therapies may not be available for a decade or two, maybe several decades. And I noticed that a lot of progress has been made on the biomems front in terms of mechanical or micromechanical ways to address some of these issues. I'm particularly, for example, there's artificial blood replacements, respirocytes. There's insulin-generating micro machines that you can insert, and they've shown quite a bit of promise. I'm curious, what is your perspective on our ability to address cancer and other age-related issues with mechanical means in the next couple of decades?
I think the role of non-biological approaches to addressing biomedical problems, whether they be problems associated with aging or indeed other problems, the role of such things will undoubtedly, inexorably increase over time. Where I really have no real feel is how fast it will go, or indeed whether it will be fast enough to outpace the more, if you like, traditional biotechnological approaches. Therefore, what I think is it's a damn good thing that all of these approaches are being tried, because the more approaches we try, the sooner one or other of them will actually work.
So if I understand right, your proposal requires that the stem cells that have the telomerase gene have to be killed by chemotherapy. If you have a tumor already started, when you start this therapy, even if it's way too small to see, then that tumor could still kill you. So you have to apply this proactively. Is that correct?
More or less.
And
is that your question, or is that the preliminary to your question?
well, that's the first half, that it has to be applied proactively. The second half is, if I understand correctly, this would cause sterility in men. Is that correct?
Maybe. Okay, I'll answer both of those questions. Let's do the sterility one first. Well, it would certainly cause problems with the normal process of spermatogenesis in the end. But spermatogenesis, of course, starts from spermatogonial stem cells, just the same as hematopoiesis starts from blood stem cells and so on. So, again, it would be perfectly possible to introduce spermatogonial stem cells that have nice long telomeres, even if they don't have telomerase genes. That wouldn't work, however, because after fertilization, right, the fertilized egg has got to do an awful lot of cell division, and it won't nearly do enough to get to, right through gestation with no telomerase. So in practice, this is something that would have to be done somatically. In other words, in order for men to have viable progeny, that would have to be done by introducing, perhaps temporarily, cells that did actually have the telomerase gene again, so that the whole thing could work. Or, of course, we could just give up the natural reproduction approach and just do it all by cloning, but you get the idea. So back to the question of whether it has to be proactive. It would certainly be better if it was proactive, and in the fullness of time, when these therapies, if they get developed properly and they become safe, I think that will certainly be the way that it's done. But in the short term, especially when the therapies are very experimental, I think the trade-off will be such that it will be better to apply them only to people who already have a short-term problem, namely they already have cancer. And the reason that could be done is, excuse me, yeah. So it turns out that you can engineer stem cells so that they are unusually resistant to particular types of chemotherapeutic agent. Not the sort of thing that you would expect to be useful, but it turns out it really is useful. I'm just going to show you an example from this guy, Les Furban, who actually died a couple of years ago, but his work is still continuing in England. There's an enzyme, well, not really an enzyme because it's not a catalyst, but it's a protein that repairs a particular type of DNA damage called alkylation of guanine. And it's possible to essentially saturate the activity of this protein by giving it a chemical that can basically, that the protein recognizes as the damaged base of the DNA. And you put this chemical in large amounts, and it just, eats up all of this stuff, and the cell can't make enough of the protein to keep up. So that means a lot of this damage in the real DNA does happen so much that the cancer cell dies. Okay, and so that's why it's a good trick. Unfortunately, it's relatively important that this not be happening to normal cells. And so the trick here is, it turns out that there are mutations in this protein that mean that it still recognizes normal DNA that's been alkylated, just as well as before, but it no longer recognizes this substrate, this pseudo-substrate, this chemical that you were putting in to trick the enzyme. So you can make your stem cells with this mutation, and this stuff, the pseudo-substrate, 0,6-benzyl-guanine, no longer depletes the protein. So the protein can still carry on doing what it's supposed to do to the DNA. If your stem cells have this mutation in their version of this gene, then they can survive a much higher level of the pseudo-substrate than a cancer would or than a normal stem cell would. So what you would do is, first of all, you'd introduce these stem cells, which had this mutation. Then you put in a boatload of the carcinogen, sorry, of the chemotherapeutic agent. It kills the cancer at better than you could survive with if you hadn't had the stem cells that had been engineered. And B, it nicely kills the stem cells that have telomerase, the natural stem cells that have telomerase. So it has two a double whammy benefit from WILT's point of view. It gets rid of the stem cells that you don't want, and it also gets rid of the cancer you don't want.
So sort of Roundup-ready stem cells.
More or less.
But
Yeah, so this is a brilliant idea. It's been around for a little while. Of course, it turns out, as with all biology, there are problems with it. It's not really quite ready for prime time, but it's nearly there. And I definitely want to. This is definitely on my hit list, high on my list of things that the Methuselah Foundation needs to fund as soon as it possibly can.
Any more questions? Okay, speaking of Methuselah Foundation and funding, here's your opportunity to pitch it.
Fair enough. So yeah, I mean, I guess I pitched it at some length in my talk a month ago, which of course is online already. So I won't go into too much detail now, but essentially the Methuselah Foundation is focused on promoting and also sponsoring research to make not only WILT, but also the other aspects of the strategies for engineered negligible senescence a reality. A lot of the work that needs to be done is already being done, especially in the stem cell arena, but a lot of it isn't because it's generally considered to be a bit too ambitious to be attractive for normal funding from the government or from industry. So we're keen to support this stuff ourselves, and we are building up as much of a donor base as we can. Of course, we're spending money as quickly as we get it because the scientists who really want to do all this work in these various areas are definitely ready to go. They're ready to roll. They just need the funding that it takes to make these things happen. I have a list of well over a dozen projects that we really need to fund, and I know who to fund and so on. Eventually, when we get to a sufficiently advanced size, we will become a more traditional funding agency which actually solicits grant applications. We're hoping to be able to do that within a year or two. But certainly, the faster we grow, the sooner this work will start being done, and the sooner we start, the sooner we'll finish. So I obviously would very much welcome any donations to the foundation, which are easy to make. My website is very easy to find, emprise.org, methuselahfoundation.org, and you can start from there. Thank you.